Hello and good evening and welcome to a very special recording of Future Proof on News Talk 106 to 108 FM. This is Health Risk. My name is Jonathan McCrea. Tonight we're going to go on a journey through the genome to find out not just what our genes and DNA can tell us about our history, but also what it can tell us about our chances of getting disease in the future. This journey, I suppose, started when I was just a little boy. My grandmother lived in a granny flat at the back of the house, and sort of done thing in the 70s and 80s, I think. I don't have a huge memory of my relationship with her, but I do remember she used to force feed us mushed banana sandwiches. But as I got older, I noticed that she'd start to make mistakes, the sort of mistakes that as a little boy, even I knew grown-ups don't make. She called me by my father's name, Billy. She would talk about events that hadn't happened, at least not in my memory, and she'd think we were in a different place to where we were. And I realized at that point that there was something very wrong with my grandmother, and she died when I was about 7. I now know that she suffered from Alzheimer's, and tonight, as a result of genetic testing, I'll find out if I have an increased risk of getting this disease. So about 8 to 10 weeks ago, I spat into a plastic container that I got in a box like this, and I sent off a sample of my DNA to an American company. And for $99, anyone can do this. You can get a rundown of various genetic traits, and a lot of these genes have a relationship to disease. So we'll be talking about that, but not just that, we'll be going through the entire process of getting a genetic test. We'll have a Q&A session afterwards, and you're very welcome to ask your questions. You can also see how I react to the results as I get them. I must stress, I don't know the results myself. My clinician here is Shane McCrea from Belfast City Hospital, and he'll be giving me the results, and you'll be finding out at exactly the same time as I will. And so you'll see on this screen here, this is a company in Clonshaugh here in Dublin. They're doing great work with biosensors. So I'm hooked up to a machine that gives you a live reading of my galvanic skin response. So you'll see my emotional response to the sort of information that we're getting here today. So that's adding a bit of drama. Okay, so we're going to start off with a question that we might ask again at the very end of the program. And I'd like to see a raise of hands from the audience. So if right now you could find out your chances of getting a serious disease, would you want to know? Raise your hands. Would you want to know if you get a very serious fatal disease? Okay, so about half, it seems. Maybe a little bit more than half. We're going to ask that question at the end of the evening, so it'll be interesting to see people's responses. Okay, let's start off with a bit of theater, shall we? So you all received a pack as you came in. You can open that pack now. Be very careful because there's 3 things in it, not 2. We're looking for the playing card, so you're all handed a playing card, and one of you was given a joker. Can the person who got the joker please stand up? Hello, sir. Do you want to come down to the stage, please? Give him a round of applause, please. What's your name? Ian. Ian, come on. Okay, so what we're going to do now is we're going to extract Ian's DNA. So this is a very simple process. We'll get producer Laura Dungan to come up on stage and do that for you. So people might not know how amazing this molecule is. Discovered in 1953, the structure of it by Watson and Crick, 2 scientists who realized that this is how living organisms manage to copy their information from one generation to another. So what we're doing here... Sean, is that right? Is that your name? Ian. Ian right now is rinsing water through his mouth, and he's scraping off skin cells that we'll use to extract the DNA. So he's got to do it quite vigorously for 30 seconds. Well done. Give him a round of applause. Well done, Ian. Okay, we'll let you go back to your seat. Okay, so we've taken the cells. They're now in a mixture of water, and what we're going to do is we're going to use a detergent to break down the cell walls that hold the DNA, and everything will spew out of those cells, and we'll have a mixture in there. And using ethanol, we'll get the DNA to clump together, and you'll be able to see this magic molecule with your own eyes. We'll show you how the DNA actually collates in this ethanol solution. So it's quite an incredible molecule, DNA, and it is responsible for the many differences between you and the person sitting beside you and us and chimpanzees. We share 50% of our genetic information with a banana tree, which is kind of crazy to think about. But we're going to start off now by going through what everyone goes through when they get a genetic test. So if you are someone who has a family member who's been through a particularly difficult and, I suppose, serious genetic disease, then you might want to get tested yourself. And to do that, you need to undergo what's called genetic counselling for the most part, and it's in your interest to do so. So joining me now on stage is Tara O'Neill. Tara is a genetics counsellor from Belfast City Hospital. Please give her a round of applause. Welcome to the programme, Tara.
Thank you.
Let's start off with the very basics. What is a genetic test?
Well, similar to what you saw, you can get DNA from any cell in our body. So you can get it through saliva, which you've just all seen, or what we would normally do in clinic is actually take a blood sample. It's cleaner. Labs are set up that way and hospitals are set up that way. So the actual genetic test or the sample is quite simple to get, but it's what that all means for the person and the family.
Okay, so it's sent off to a lab and they run it through a computer.
They do all their magic. And
then we got some magic stuff. We might talk about that in a bit. Yeah. But your role as a genetic counsellor is very interesting to me. So what exactly is it that you do?
I will, as you mentioned before, it would normally be someone who's had a significant family history of a condition or genetic disease that would come to us. So I would go through their family history, see what the condition is, how it's inherited, and we'd have a diagnosis already. And if we have a genetic test that is available, unfortunately they're not available for every condition, but if it is available, then we can offer that test to the person, and that has a lot of implications, to them and to the family.
So what sort of diseases are we talking about? People wouldn't come to you and say, my brother has red hair, I want to know if I have the red hair gene.
No. But it would be serious things. Breast cancer would be a big one. We've all sort of heard about Angelina Jolie in the media lately. So that would be a big part of what we would do. If someone has very significant family history, not breast cancer is common, you would need to have quite a significant one. And then we would go through the family history and test them for the BRCA1 and BRCA2 genes and talk through what all that means for them and implications. Yeah,
I have to say the coverage of the Angelina Jolie story was actually pretty good, and it really brought the idea of genetic testing to the public in a way that we haven't really seen before. Angelina Jolie had this history of this particular disease in her family, and so she decided to go and get a test, and she found that she was positive for this gene. And this was a gene that can suppress tumors, and if there's a mutation there, then it doesn't do that particularly well. And so she decided she was going to have a double mastectomy and have her breasts removed as a preventative measure. Yeah,
it's something that some women decide to do, and we sort of calling it the Jolie effect. We've gotten a lot more interest in breast cancer, so she's done sort of a good job to put it out there and make people aware that testing is available, and not just testing, but there are options, which is a lot of what we would sort of talk through women. It's yes, you might get this bad news result, but there's something we can do about it. Screening, surgery, and that's a lot of what we would do in genetic counselling.
Because obviously not a lot of people, some people get that to 80%, and depending on where they are in their life, they might decide to have their breast removed or not.
Yeah, very different. And you might have 3 sisters that would all come up to you and they'd all come get this test, and they'd go, one person might be, under no circumstances would I even think of having my breast removed. That's drastic. And some people think, why would you want to do that to yourself? But then you might have someone else who thinks, well, my mom died at 40 of breast cancer. There's no way I'm going to, have my kids grow up without their mom. So there's no choice about it. I'm going to have my breast removed. So people are very different. So it's interesting to see where the people come from.
Well, I suppose, in some ways, at least you can do something about breast cancer if you're armed with that information. But of course, some people come to your door and their family member has an incurable disease, and that's a very different and sort of murky world, isn't it?
Yeah, it's one of the sort of big examples that we would use is Huntington's disease. I'm not sure if most people have maybe not heard of it, but if you have the gene for it, you 100% will get it. There's no treatment, there's nothing we can do, and it's a neurological condition, and it's degenerative, and there's just nothing we can do. So it's for someone coming to genetic counselling to get that testing done, there's a very strict protocol we would go through. You'd see someone at least 3 times before you would take a blood sample and do the testing. Because once you have that information, there's no going back. And there's a lot to think about before you would do that.
Yeah, I mean, essentially you're looking death in the face. If you go to get this test, you sit down, and you were telling me earlier that you don't just go, well, my family member has Huntington's, I'll get the test. You have to go through a number of counselling sessions, and that's to presumably protect the person from the massive news that they could have a fatal disease.
Yeah, the last thing we don't want is for someone to get that result and be left in this sort of despair. And we want to make sure that they know exactly what this means. And we normally say, well, come with someone. It's important to have someone to go through the process with, so you can talk it through with them. You've got support, because when we give you that result, there's just no going back, and you're left with that, and you're left with the implications that has for your children. How do you tell your children? Because if you have it, it's a 50% risk to your children having it. And if you're coming before you have children, do you then decide to have children? What
so people actually make decisions based on these genes as to whether or not they're going to have children.
Yeah, they would, and there are options that are available even if you do have a genetic condition to have children that are healthy and not affected. But still, people do make decisions based on it, and that's a lot of what we would talk about is sort of options. There's so many implications to getting this sort of information, isn't it? Which is why there's so much counselling that goes on, because it's not just the person who has the likelihood of getting this disease, it's also their family members, and there's a thing that counsellors call survivor guilt. Yeah, sort of used example before of three sisters that might come up for testing for BRCA, the BRCA1 mutation that Angelina Jolie has. If they have a family history, if say their mom has had the gene, they're at 50% risk each of having the gene, and they're not all going to get the same results. Some might come back having the gene, some might come back not, and something we would talk to them is to think, well, if you don't have the gene and your sister does, a lot of sisters would feel quite guilty. Oh, I wish I had it and my sister didn't. I could cook better than my sister. So it's not all good news. You'd be surprised the amount of emotions that sort of go into it.
So, in that case, it seems like there's no winners. Even if you don't have the gene, you're still sort of stricken with guilt because you were the lucky one who avoided the...
Yeah, and then you have to sort of see your sister or, other family member going through the pain of having it.
What is it like giving this sort of information to people? How do people react? Do people really fully understand what it means when you say, well, you've got a 50% chance of having a disease that will kill you by the time you're 40 or 50?
It sounds very dramatic.
No, it does.
But it is. Yeah. Well, the whole point of genetic counselling is to prepare someone for that result. I'm not a counsellor. I'm not trained in counselling. I'm a genetic counsellor, so it's more information giving. The whole point of the whole process is so that they are ready. And people don't, people hear about genes and DNA, but, what's the inheritance and the different conditions of different patterns of inheritance. So people don't always just know. We can't expect people to know how something's inherited and what their actual risk is. So that's what my job is to do, is to sit down and go, right, so, your mom had this BRCA gene, so then you're at 50% risk, and why you're at 50% risk, not just saying it, explaining why, about genes and how they're inherited. And the experiences, normally people go away going, oh, okay, right, I get it now, I understand. And when they do have their results, they should have that knowledge, and it shouldn't be so much of a shock, and that's the whole point and what we are trying to do.
Does anyone come to you for counselling and regret getting that test, regret opening Pandora's box?
The whole point, as I said, is to try and not have that, but yeah, it does happen, and you're going to experience tonight what it's like to just get a result. So maybe be able to relate to that sort of feeling of anxiety and worry, or maybe not that I want to worry you, but
Well, I'm hoping for the purposes of this evening that there's no drama. I know everyone would love. Anybody actually want to see if I get a really bad result?
Nearly, but people do quite. We don't want that, but yes, there are people that sort of go, oh, I wish I didn't know. I don't know how to deal with this information now, because it is, it is serious information. Knowing that you're at risk of developing a disease, so it can happen.
And you can't put that genie back in the bottle, essentially.
No, that's why it's about informed consent, making sure they don't just give a blood sample, get a result. It's you need to know what you're getting yourself into, because as you say, you can't put that back.
Tara, Neil, thank you very much. Okay, coming up on the programme, we'll be getting into the results as Kevin Mitchell from Trinity College Dublin will be telling me about my percentage Neanderthal and also my likelihood of becoming an Olympian sprinter. Don't go anywhere. You're very welcome back to Future Proof on News Talk 106 to 108 FM. This is a very special event. It's Health Risk Live at the Science Gallery. And tonight I'm finding out the results of a genetic test I took 10 weeks ago for the princely sum of $99 plus postage. This is a test that anyone can get, and it will tell them about their likelihood of getting certain diseases, but also some other interesting traits, which a friend of the show, Kevin Mitchell, a neuroscientist and geneticist at Trinity College Dublin, is here to deliver. Welcome to the program, Kevin. How are you?
Much. Very well, thanks.
So talk to us about traits and inheritance and how that works.
Well, so DNA is basically the information that all living things carry to copy themselves, as you said earlier. And so every fertilized human egg has inside it the DNA to make a human being. And more than that, every cell that you have has the DNA to make not just a human being generally, but a person like you. So not exactly you, because there's more to you than just your genes, but effectively your identical twin. Okay. So the DNA code, that information is encoded in these 4 letters of DNA, the A, C, G, T, which occur in a specific sequence in the genome. And it's that sequence, whether it goes AAG or AAC, that carries the information that affects the traits that we have.
So we have 6 billion of these letters in our genome. 3 billion pairs.
That's right.
And across these letters, every once in a while, there's a different letter swapped out. And these are the things that make us different, make one person tall, one person short, one person have red hair, and one person not. Is that right?
Exactly. So if you look along the human genome, you could say, you can think of it as just a linear sequence from start to finish of 3 billion letters long. And say you start at position 1, the sequence might go A, A, G, C, T, A, A, T, something like that. And that would be pretty much the same in practically everybody on the planet.
Because we share 99.5% of our
effectively 99.5% of your sequence, and my sequence is the same. But it's the 0.5.
Samesies. High five.
Nice. It's the 0.5 that's different. That's the key thing. And as you mentioned earlier, we share 50% with a banana and 98.5% with a chimp. So we're not that much more similar to each other than we are to a chimp. But so if you go along that sequence, you could go A, A, G, C, T like that. But then every once in a while, there's a position where some people have an A, some people have a T.
Yeah.
So what we call a variable position. And about 1 in every 1,000 of those letters will be a variable position like that. And so what we can do is actually get a genetic profile. What this test does is actually get a genetic profile that looks across your genome at all of those sites and says, okay, at the first variable site, Jonathan has an A. At the second one, he has a G. At the third one, he has a G. At the fourth one, he has a C. Whereas I might have a T and an A and a G and a C. And so that profile of the differences is really what this test gives you. And those differences are important because they affect, well, first of all, those differences are inherited. Okay, so we can use them to track ancestry in the first instance, and they're extraordinarily powerful to do that. So those are the kinds of things that would be looked at if you did a paternity test.
Yeah.
And to do that, because you're looking at a very close relationship, you only need to look at a few markers.
That's, by the way, a very thriving business in Ireland. I don't know if you know that, but paternity tests, it's a business to get into if you're interested in tonight's talk.
So, but what this test does is not just look at a few markers, it looks at about 500,000 markers across the whole genome. So you get a really, really dense, detailed profile. Because of that, you can look not just back 1 generation, you can look at relationships back hundreds of generations into the past. And we will do that when we get the results from your ancestry. We'll look to see what part of the world your DNA derives from. We'll look at the inheritance of your Y chromosome, which is passed just down the male line, and because of that, it gets a different kind of a signature to all of your general DNA. We'll look at another piece called your mitochondrial DNA. So there's most of your DNA in your cell is in what's called the nucleus, but there's another little part of the DNA called the mitochondrion, which has its own DNA. Another little part of the cell has its own DNA, and mitochondria are only passed on in the egg. So they only come down the maternal line. So we'll have the Y chromosome to look at your paternal line, the mitochondrial DNA signature to look at your maternal line.
Okay.
And so we can go back in human generations into history, and actually we can go back even into prehistory to see, like you mentioned earlier, how much Neanderthal DNA you have.
Yeah, which I'm sure is hardly any. So these changes, and I think this is really interesting because people talk about, we have different changes in our genes. This single letter can sometimes be the difference between a healthy person and a sick person. So when we talk about, someone has a gene for something, we mean that one of those 6 billion letters is different from everybody else. So say, for example, cystic fibrosis is a perfect example of this,
That's right. Yeah, so cystic fibrosis, which happens to be very common in Ireland, is caused by a mutation in the genetic code. That part of the genome codes for a protein. So that's what genes do. They make, they're basically a recipe for a protein, and all the bits of your cellular physiology are carried out by different proteins. They're like little robots or machines in your cells. This particular one sits in the membrane of cells in the lung, and it pumps chloride in and out of the cell. And if it doesn't work well, then the lungs get gummed up, basically.
Yeah.
And so the mutation, what we call the gene for cystic fibrosis, is really a mutation in that gene for the protein that makes that chloride channel. And that one mutation will cause that disease. And it's an example, like Huntington's disease, of a very definitive, extremely high probability, effectively 100%, that if you have that mutation, you will have that disease.
It just goes to show how incredible our bodies and how every living thing is at passing this information on. When we start off as a fertilized egg, we separate over and over again, and yet the information, the DNA in our cells, is copied so perfectly over and over again that every once in a blue moon we have a single letter mutation in 6 billion. And actually, I spoke to a guy on the program before, and he was talking about using DNA as a storage device, that actually we could store information on DNA in the same way as we do with a USB stick. And he said if we were to use it in that way, and I know that's sort of blowing your mind a little bit, but if we use it in that way, it would be the most stable storage facility. The problem is reading and writing takes forever, so you just be there with your USB stick going on, for God's sake. But this is an amazing mechanism for copying and copying and copying, and not just cells, but of course the cells that make us people. So generation to generation to generation. That's the magic of DNA. So let's talk a little bit about traits. And we were talking about how traits are inherited. We're going to find out a little bit about your DNA now. So open up your packs. You'll find there's 2 things inside the pack, okay? One of them is a mint. You might have that at the ready. And the other is a paper strip. I'll give you a couple of minutes to get together. So what we're going to look at here is your ability to taste the bitter compound. And we put a kind of a user-friendly name on it. We've divided, when I say we, I mean scientists have divided people into tasters and non-tasters, okay? So some people have an ability to taste bitter, this particular type of bitterness in a very strong way. Some people won't taste it at all, and some people will be around about middling. But about 30% of you won't have any reaction. Some of you may have a rather extreme reaction, but don't worry, you're not damaging your health. So when I say now, because we want to get everyone at the same time, when I say now, we all ready? I want you to put the piece of paper on your tongue for 5 seconds, okay? 1, 2, 3, now. Okay, so we're seeing various reactions to some people. Anybody finding that disgusting yet? Okay, 1, 2, 3, 4, 5, 6, 20. Oh, actually, do you know what? Not that many. That's quite a low amount. Anybody else find that really bitter?
Oh, there's more coming up.
It's taken a while for some people. Maybe you've got a bit of a dry mouth. So it depends on different variables. But so there's a percentage of you. Anybody not taste anything at all? Raise your hands. Wow. That is, I suppose that statistically would be a bit of a wobble, but people who have, obviously, if you taste the bitterness, the mint is to get rid of that taste. There's nothing scientific about it. So there are some people who have a specific copy of that gene. Some people who have 2 copies of the gene, and they'll taste that bitter compound in a very major way. And then there's people who just have 1 copy of that gene, and so they would just be like regular tasters.
Yeah, so it's an example of how genetic differences can affect a trait. In this case, it's perceptual. And another very common example of that would be color blindness, where there's a mutation in genes that detect various wavelengths of light in the eye. And that's extremely common in males. Maybe 2 or 3% of males can be red-green colorblind. But those genetic differences will affect all kinds of other things. So, like your eye color, your hair color, how tall you are, even how heavy you are. So lots of physical traits, but also psychological traits like intelligence, personality traits, and so on. And also risk for various diseases.
Okay, let's do it. You have some results for me.
I do have.
So this is, as I say, this is the first time I've heard about these results. It's been painstaking to try and make sure that that I don't know these results, and also that that we actually do get the right information at the right time. So thanks everyone for the secrecy involved. Okay.
So
open the envelope.
Open the envelope, and so what we can do to begin with, as I said, one of the really powerful things about this genetic information is you can use it to trace ancestry. You can use it to find relatives. You can and look back in your own past, which is what we're going to start with, and then we're going to look at some of these traits that can be affected to see how well your genetic profile actually predicts your real phenotype that we see before us.
So, talk to me about my ancestry.
Okay,
was I a Viking?
Well, first of all, it's possible, but first of all, what we can do is if you look at that profile, as I said, if you've got 500,000 of these variant positions and you've inherited them from your parents who inherit them from their parents and so on, eventually what you get is actually kind of country-specific profiles or regional profiles where that this A versus T at the first position, the A might be quite common in Europeans, the T might be quite common in Asians, for example, or the G might be common in the Irish and the C common in the French. So it gets down to quite localized.
Okay, so basically you're saying that we can actually look at these letters and because they're particularly common in a particular area, we assume that that's because they were bred.
That's right.
And so that's how we trace our DNA back to certain countries. So.
Exactly. So it's like doing a paternity test, except back hundreds of generations.
Yeah.
Basically. So the overall pattern from your ancestry is probably not going to surprise you very much. It is effectively 100% European in the first instance.
Yeah.
Not so exciting.
Okay.
And then if you go down a little bit closer, it's what they say is they can assign at least 50% of it to British and Irish.
What, 50% British and 50% Irish?
No, 50% British and Irish together.
Oh, I see. Harmony, beautiful.
Yeah. And so there's 43%, or most of the rest of it is what they call non-specific Northern European, which doesn't mean it's not British and Irish. They just can't tell. Okay, so
what?
So effectively, it's
how much money
British
do I pay for this? It's rubbish. Okay, so I'm a bit British and Irish, and then something else.
Well, no, I mean, there's something else could still be British and Irish. It's not that it's not.
Okay, so they know nothing. Let's move on.
Okay, nothing new anyway. No surprises there. Okay, so the next one then is your Y chromosome. And so you can track Y chromosomes far back into the past because of the way that they're inherited. They basically just get passed from father to son.
Yeah.
And so it will only go back from your father's father, his father, and so on. So you're looking down a very select, just one angled a glance at your entire pedigree.
Okay.
And as that happens, they can accumulate mutations. And we can track those mutations that are then typical of certain populations.
Okay.
Sort of similar to the ancestry thing.
You're going to tell me I'm British and Irish a bit.
I'm going to tell you you're British and Irish, exactly. So your Y chromosome type is first of all, very, very common in Northern Europe. And it reaches its height in Ireland. So it's actually a prototypical Irish Y chromosome, which may be good because this can be an inadvertent paternity test. Which so
you're half Japanese, mommy.
Hopefully that's not a surprise to you. So this particular one goes back, and in fact it's extraordinarily common in the Irish population. I have it myself.
Okay. Samesies. High five.
Nice. So one of the interesting things about this, actually, the history of it is that it's so common in the Irish population that you can think there must be a reason for that, and there must have been some person where these mutations arose, or who carried those, who had a lot of descendants in the Irish population.
Sounds like me.
Yeah, and so for
a Lothario type.
Exactly. In fact, possibly a Nile of the Nine Hostages type. So this is the theory, at least, that because this particular Y chromosome is really diagnostic, actually, or very common in people with the surname O'Neill, like Tara there, except Obviously, she doesn't have a Y chromosome. But so people with that surname who can trace ancestry, at least apocryphally, back to Nile of the Nine Hostages, have that Y chromosome and many other clans that are related to that. So you can look back in history and trace surnames which go along in Ireland, at least, with the Y chromosome.
Wow.
And back to Nile of the Nine Hostages. And the idea is that because he was a king and he had lots of opportunities for conquest and he had many sons who had lots of opportunity for conquest, that may explain why his Y chromosome eventually came to dominate the landscape in Ireland.
Yeah, so you could say to anyone in Ireland, who's your daddy, and almost certainly it would be Nile of the Nine Hostages.
Exactly.
Okay, so let's move on.
Okay.
Are we going to talk about my ability to be an Olympian yet?
Not yet. We're getting there. Okay, so we're going to talk about the maternal line, which is your mitochondrial DNA. And this one is a little different in that the type that you have for mitochondrial DNA is very highly concentrated in Eastern Europe and the Near East. Not so much in Northern Europe.
Okay.
So now...
This I got from my mum, and my mum got it from her mum, and her mum got it from her mum.
Right, but that could be traced back. This could be coming into your pedigree from 10 generations back or 100 generations back.
So roughly, what's the border here?
Well,
countries change all the time.
Yeah, I mean, it's basically right in the Caucasus, around the Black Sea, that sort of area. And but what it does say here is that it's sort of concentrated in Turkey, but this particular mitochondrial DNA variant has made its way across Europe in 2 separate passes, eventually reaching the British Isles. So at some point, some woman with or a group of women who had that type migrated through. And the interesting thing is you can track human migrations by looking at the frequencies of these various types of Y chromosome, types of mitochondrial DNA. You can infer human history and look back and say, okay, there must have been a migration from here to there. One of the interesting things with your Y chromosome is it's quite common also in the Basque country. So some kind of cross-fertilization maybe, in history. There.
Okay, very good. Okay, so is this because I'm really interested. So there's a particular gene that is very common in those at the very top of athleticism. So the world's best sprinters, it's very common, the world's best cyclists and so on. And it's almost unheard of to have this particular gene and not and be in this sort of group of elite athletes. Is that right? Almost every Olympian has this particular gene. So this is the gene for success in sport.
Yes. Now, do you want to hear about that or do you want to hear about caveman DNA first?
Is it caveman DNA? You're going to tell me that the caveman was in Northern Ireland or something?
I'm going to... Okay,
come on.
So by tracking these human migrations, like I said, we know that actually when modern humans emerged from Africa, they emerged into a Europe where Neanderthals had been for 100,000 years already. And the idea was that they outcompeted them, and clearly we did. But there was always some question about whether we might have actually made it and intermingled with them at some stage. And in fact, there's now clear evidence that we have, because we had sequenced DNA from Neanderthal remains, and we can see now some remnants of that in our own genome. So the average percentage of the genome that can be traced to Neanderthals is about 2.7%.
So most people, you're 2.7% roughly.
Yeah,
Neanderthal,
that's the average.
You're 2.7.
Yeah.
I am
5%.
Oh, 5%?
No, not really. I'm just kidding. You're 2.8%.
Yeah, it's still more than the average, though.
A little bit.
5%. I was thinking, how could it possibly be 5%? I thought the variance was like a couple of percentage points. So, okay, you scared me there for a minute.
Sorry.
So I am a little bit more Neanderthal, which means that I'm not as good with tools. My forehead is a little bit like that.
Got a little thing going on.
Shut up, Kevin. Okay, I want to hear about the Olympian gene.
Good. All right. So there's a bunch of traits that they can sort of make predictions on based on your DNA. One of them is the sprinter gene, and actually what it says is it's called muscle performance, and the outcome is that you're a likely sprinter. So is that...
A which?
A sprinter.
I'm a likely sprinter?
Yes. So
in what circumstances?
Well, I presume if you're running fast.
If I wanted to. You're a likely sprinter. Is that what it tells me? It doesn't tell me I could be a world-class athlete?
What, do you want a 100-meter time?
But like there's a specific gene that's found in these Olympians. Do I have that gene, that variant of it?
I think that's right, yeah.
Okay.
Now, that doesn't mean... So that particular variant is found in almost every Olympic sprinter, but it's also found in lots of people who aren't Olympic sprinters, obviously. So it's a kind of a requirement, but not necessarily going to be enough to make you Usain Bolt.
There's an interesting kind of aside to this, and that is that while the studies have looked into the presence of this particular gene in people at the very height of athleticism, there is one person, I think it was a pole vaulter or a long jumper, someone from Spain, and they actually managed to reach the height of their field. Even though they didn't have this particular gene. So against all odds, they managed to fight against nature and become the best of the best. So you can do it if you haven't got that gene.
So some of the other ones on here are interesting. So eye color is very strongly genetic. It's basically 100% genetically determined, but it's a little bit complex. So what they predict here, just from your DNA, is that your eye colors are likely blue, which is...
It's green.
It's green. It's
greeny
true.
brown.
Yeah.
Couldn't be further from blue.
Well, it could be browner. But
why is brown any less blue than green?
Because that's what green is.
Slightly. That's my Neanderthal speaking.
Exactly. So it looks at hair curl. It said your hair is slightly curlier hair on average, which I think is
it is slightly
fair enough.
I got that from my mama, who's over there. You can see that big woolly mop of hair.
Okay.
She's got all of those genes.
Well, unfortunately, it's also increased odds of male pattern baldness. So you might not keep that curly hair. We'll see.
Really?
Yes.
I was always so sure. My dad, look at my dad. My dad is stand up, dad. Stand up, dad. Stand up, dad. He's a good-looking man. I thought, I know, because people are going to say, oh, you get your hair, your baldness from your mom. Not just your mom, that's a bit of a myth. It's mom and dad.
All right.
So, okay, so that's not
Okay,
good
so
news.
let's see what else. It says you can taste bitter stuff. I noticed that you didn't volunteer to taste that nasty tasting thing.
It's disgusting.
Your earwax type is likely wet. Which is not
And
going
how
to...
does that, why, how does someone come across that information? Why,
Well,
who cares?
It's a genetic trait, and they know what the variants are that cause it. So
hang on, you tell me
is.
someone age 12 went, oh, I'm going to study really hard. Oh, I'm going to learn all these things. Oh, I want to do science. 18, oh, I'll go to college. Oh, learn all this. Spend 7 years doing, and they get to the height of their career, do a PhD, and then they go into research, and their discovery was some people have wet earwax and some people have dry earwax. Genuinely.
yes.
Wow.
Well,
Okay, what else?
The amazing thing is they got a grant to do that.
Okay,
so
what else do we have?
So you're likely lactose tolerant.
Yeah,
Yeah, good. Let's
so.
see.
like my milk.
If you were a smoker, you'd be likely to smoke more.
Oh,
Are you a smoker?
I used to smoke. I don't smoke anymore.
So now you smoke less.
Yeah.
Okay.
So we're about 50-50. I mean, if we rolled a dice, and
Yeah.
would I save myself $99 is a question.
It's possible. Well, I mean, so here's another one which is more serious, which is actually resistance to HIV/AIDS.
Oh.
So there's a mutation in a protein that is the protein that the HIV virus uses to get inside white blood cells. So it's a protein that sits on the surface of your white blood cell. The HIV virus grabs a hold of it and basically uses it to get inside the cells.
And that's the problem with HIV. That's why it's so difficult to get rid
Exactly,
of, because it gets
hides inside there.
your cells. Yeah.
If there's a mutation in this protein, the protein still works and does its job normally, but the bit of it that the virus recognizes is changed and it can't get in. So it's quite rare. It's about 5% of the population carry that mutation, and you don't.
Oh.
Sorry.
Tell me the interesting stuff, Kevin.
Well, those are all the ones that come up with a positive result. And I mean, one of the things that they have on here is what they call a kind of a confidence estimate of how much confidence you can have in that prediction. So the ones I just told you were the ones that they make what they call a confident prediction of. We've seen that maybe they didn't all hit the mark 100%. There's a whole load of other ones on the list of things that they look at that didn't come up with a confident prediction for you.
Yeah.
They might come up with a prediction for somebody else.
Okay, so we might go into some more of those kind of traits a little bit later on. If people have any particular questions, I know there are some people who emailed the show before and say they're coming along, they'd like us to ask them questions. So we might go into Q&A and talk a little bit more about those sort of traits and how they might apply to the general public. But I think we'll leave it there because I know people will probably want to get into the meaty stuff. At least it's all building up to this. So Kevin Mitchell, thank you very much. Stay there and you can join us in a sec when we have Shane McCrea on. So in a few minutes' time, this is it. I'll be finding out my likelihood of having an increased risk of diseases like Alzheimer's, Parkinson's, and breast cancer. Don't go anywhere. Welcome back to Future Proof on News Talk 106 to 108 FM. I'm Jonathan McCrea, and it can't have escaped you that we are out of the studio. We are at a live recording of the program. It's called Health Risk. It's a part of the Risk exhibition here at the Science Gallery, where I am finding out the results of a genetic test I took about 8 weeks ago, where I paid $99 to find out some of the secrets of my genome. And here to deliver these results, and I've mentioned it before, but I haven't heard these results before this very second, so you'll be hearing them live as I do. Here to give you those results is Dr. Shane McCrea. He is my clinical geneticist, and he's from Belfast City Hospital.
Yes, good evening.
Good evening. We talked a little bit about whether or not people should know this sort of information and what sort of effect it will have on you. As a clinician, do you deliver these results a lot of the time?
That's right, Jonathan. We have patients that would be coming up and they'd be concerned about a family risk of something, and perhaps we'll already have made a diagnosis in the family and have got a specific genetic variant that we've been able to associate with that condition, and then we would see other members of the family who may wish to be tested for that to see if they would have it themselves, and we would deliver those results to them. And as Tara has explained already, that can be quite a big deal to get results like that, because it suddenly takes something that's fairly nebulous that you haven't quite clarified and tied down and makes it very real.
Yeah.
Because it is literally the opening of Pandora's box. You don't get things back in once that information is known.
Is it a difficult thing to say to someone, I've got some bad news, or is it something you get used to?
It is difficult to tell someone that it's bad news, and unfortunately I seem to be giving more bad news than good news to people these days. But I'm not saying anything about tonight, and I just want to reassure you on that. But no, we do give bad news, we do give good news. It's never easy, but what we're trying to do is to equip people, as Tara had explained, with the skills and the knowledge to actually handle the information that they're going to get. Because when we do a genetic test, we're not uncovering we're not casting the die at that stage. It's already cast. The information is already in the genome, but we are revealing that, and so it's more a knowledge thing than anything else. And very often people would come to us. They'll usually come to us knowing what the condition is in the family that they're dealing with. So the information very often that we're giving them is either going to confirm their worst fears, which they normally have themselves worked up into, or to remove those fears. So actually, in a lot of ways, funny enough, a lot of people emerge after having a genetic counselling session a lot happier.
Because it's sort of, they've spent all this time worrying about have I got it or haven't I got it, and it's almost the anxiety that's worse than actually knowing.
The uncertainty is a very big thing in a lot of this. The particular type of genetic testing that we're talking about here is called predictive genetic testing. It doesn't fully predict what's going to happen, but it's, this is the question. You're going with a specific question and you're addressing that with genetic means, and then you're giving a result based on that. And that certainty for a lot of people can be very helpful. It's not always going to be helpful. Some people don't want to know, and a lot of the counselling sessions that we undertake beforehand are to give people the assurance that if they don't want to know, it's okay for them not to know. Okay. So we don't want to push people into that. We want them to feel that they're in control of the process and that they have a degree of ownership over it.
Okay, so before we find out the results of the test, it should be time enough. So do we have Ian's DNA? So earlier in the program, we took DNA from a member of the audience. His name is Ian, I think. I'm not trying to delay or anything. So let's see. Oh, it goes. They saw it, did they? Oh.
You all saw it. It was the bubbly stuff.
See the bubbly stuff? Yeah. So that's DNA.
They have seen it.
Okay, great. Spent a lot of time figuring that out and still balls it up. Right.
If people were watching carefully, they would have seen that.
Okay, excellent. I didn't see it, but people saw it.
It was really impressive. Yeah, folks, yeah.
Okay, so let's go through the results then. So we have a couple of slides up on the screen to help you explain. So do you want to take us through my first result? What's this about?
Well, the first one that we looked at was, as Kevin has discussed with you already, the cystic fibrosis gene.
Yeah.
The cystic fibrosis gene, as Kevin mentioned, it's a chloride regulator, little channel in the lungs. It's called CFTR. And we know that in the population in Ireland, approximately 1 person in 20 is a carrier of this condition.
It's particularly popular in Ireland. Not popular, that's the wrong word. It's particularly common in Ireland.
It is, yes. And there are various reasons that have been proposed for why that's the case. But it is one of these things that genetic conditions vary in their incidence, particularly conditions like this in different parts of the world. Now, this is called a recessive condition. What a recessive condition is, because you get 1 copy of each of your genes, most of them anyway, from your father and 1 copy from your mother, you end up with 2 copies of, that's simple addition here. I think the audience is probably fairly familiar with that sort of thing, I would like to think. So you get 2 copies of the gene, and you only really need 1 copy to be perfectly healthy. As long as you've got 1 functioning copy of the CFTR protein in the cells in your lungs and in the pancreas and various other places, it can do its job and you'll be perfectly fine. However, people with cystic fibrosis inherit 1 faulty copy from one parent and another faulty copy from the other parent. The parents are carriers and they're fine, but the child will have 2 faulty copies of the gene.
Okay. So theoretically, although as far as I know, there's no history of cystic fibrosis in my family, but theoretically, I could have this gene and not know it, and I could theoretically have a child who would have cystic fibrosis, even though I had no symptoms. If
you were a carrier and if your partner was a carrier, then the chance of your child being affected with the condition would be 1 in 4, because there would be 1 in 2 chance of passing on the faulty gene from mum, 1 in 2 chance of the faulty gene from dad. So, and we know that 1 in 20 people in the population approximately have that. So if you work out the mathematics, it's not that complicated. You take 1 in 2, 1 in 2, that's 1 in 4, and then 1 in 20 times that for each one, then you end up with 1 in 1,600 of approximately in the population. It's about 1 in 2,000 we would normally quote as the underlying risk of cystic fibrosis. And the good news for you is that you don't carry any of the common mutations. There's about 50 common mutations that are tested for in this particular kit, and that would include most of the mutations, the large majority of the mutations, because each variant in the gene is different.
Okay.
Different spelling changes, different mistakes in the gene, effectively.
Great.
You can see. So the 50 common ones that we would see in the population, you don't carry any of those.
Wonderful. Okay, so that is good news. I was going to say, how about a round of applause, but that is a strange thing to give applause for. But
yeah. Well, considering that what we've got here, 150 people here.
Yeah, 150
tonight.
People here,
so there's there are going to be quite a number of people here. Kevin, do the maths for me here. I'm under pressure. There are going to be a certain number of people here that
may carry the gene, but
that will carry the gene, but be otherwise completely
may not
fine.
have cystic fibrosis. Okay, so that's great news. So that's cystic fibrosis. What's the next condition I was tested for? Because I actually don't know what condition I was actually tested for. This is such a secrecy associated with this because I didn't want to ruin the surprise for myself, because I think it's important to see this actually happening. So I don't really know.
I mean, it's a worthwhile thing to point out that when you do a test like this and for the health results that are given out, those results are locked on the result website that you
Can we bring up? Actually, no, we'll leave that for the moment. We'll do it later. We'll show people the website, but there's an actual section of this site that's locked off that the general public have to say, yes, I've spoken to a genetic counsellor. Yes, I know this is pretty heavy information. And they're the results that Shane is delivering.
So you've done the whole thing now. You've spoken to a genetic counsellor and clinical geneticist and neuroscientist and geneticist. So you've had pretty good professional input here. So I think you'd be clear to open up any of these locked ones on the site. The next one that we were going to talk about was your breast cancer or family risk.
Okay. So this BRCA1 gene that we were talking about, obviously men can have this gene and pass it on to their daughters.
That's correct.
And also men can actually also suffer from breast cancer themselves.
That's right. I mean, male breast cancer is a fairly uncommon cancer, but actually it's common enough. We do see it, and it can be significant in a family history context of breast cancer. So as Tara was mentioning, if you've got a significant family history of breast cancer, 2 or more relatives, and particularly if they're at a young age, so say 40s and maybe below, but if there's breast cancer in a male on top of that, then that can also increase the risk or the overall chance of that being a genetic form of breast cancer. There's only maybe 5% of breast cancer, 5 to 10% that is genetic in origin.
There is, as it happens, there is early onset breast cancer in my family, so this is something that I suppose.
I mean, to contextualise this though, and what this test will have tested for, Kevin has already given us some very useful information in regard to your ancestry. And although we've not really been able to classify a certain amount of your Northern European heritage, we haven't been able to identify it specifically down to the Ashkenazi Jewish population.
Okay.
And we know that the Ashkenazi Jewish population, because they underwent really quite a tight genetic bottleneck, probably around medieval times in Eastern Europe, we know that there are several areas of their genome that are very much more or much higher represented in terms of frequency in the Jewish population than they would be in non-Jewish populations.
Okay.
So
this isn't a general population result. This is a population if I happen to have this sort of ancestry history in me. So
pretty much, yeah.
And
so what we did was we tested, or the company tested for the 3 most common mutations that we would see within there are 2 of them that are in the BRCA1 gene. There are these 2 genes, BRCA1 and BRCA2. They tested for 2 in BRCA1 and 1 in BRCA2 that we see specifically, not exclusively, by no means exclusively, but specifically in the Ashkenazi population, sometimes in Eastern European populations as well.
And
so the first one is the BRCA1 185-delG, which isn't going to mean an awful lot to most of you, but it's enough to knock that gene out of action. So it's a tumour suppressor gene. If you knock it out of action, then it's like taking one brake off your bicycle. If the other brake goes, then you're into the hedge. So BRCA1 185-delG is absent.
Okay.
So you're clear for that one.
Good.
So that's fine. So 1 down, 2 more to go. So the next one is the BRCA1 5382 in C. Now
I don't like the sort of circus drama you're putting. Ta-da! You may have had.
Well, I'm not quite sure what your stress is going on. I'm going to have to ramp this up a little bit if we're going to see any response on your
That's not the point of this.
It isn't. So, I mean, when we have these, I better explain what these actually mean. That 5382, the BRCA1 is the name of the gene. The 5382 tells you the position, the precise position within the coding sequence of that gene. So this is a little letter that specifies an amino acid in a protein. So this is the TCAG. And INS means an insertion of a C. So it's a little letter C that has got stuck in there, and then the rest of it's all shunted down. The del that we had previously is one that's deleted and taken out. Now, if you move a DNA sequence 1 or 2 to the left or right, because your body is reading these in groups of 3, and each triplet has a certain code and codes for a specific amino acid in the protein. If you take one out, then that's what's called a frame shift. It knocks the whole, what's called the reading frame of that DNA molecule or of the sequence out. So it means that the protein basically eventually runs into the sand and just can't be produced. So the BRCA1 5382 in C is
absent
again. So that's great. This is good news.
I love the drama. Good.
It's actually, this is specifically important because remember, these are inherited variants. These are not little probabilities, they're sort of, we're not putting your risk up a little bit or down a little bit. These are pretty major things. So if you had either of those, then that would be significant in your family history context. Probably not so much for you because you're a male, have been well briefed beforehand. So you're not going to be likely to have this sort of a problem, but if you had daughters, that would be a risk.
Yeah.
It could be a risk for the wider family, and obviously mum's here as well today, so that's the kind of thing that we would need to explore then within the rest of the family. So the next one anyway is the one on the BRCA2 gene. Okay. And it's the BRCA2 6174-delT, so you know what that means.
Yeah.
Number 6174 little nucleotides into that, there's a deletion of the T, and it's absent as well.
Okay, so, phenomenal. Okay, so that's good.
So your Ashkenazi Jewish mutations are not there. The problem with this is not going to give you the all clear from the BRCA1 and BRCA2 point of view, because this is not a full sequence of the gene.
Yeah. These are specific mutations for a specific population based on what we know. So these aren't actually the Angelina Jolie BRCA1, BRCA2 tests because
they're not.
They cost a lot of money. Yeah. And actually, one of the things about this website is it does say it tests for this, and people might be misled into thinking, oh, well, I'm told by my clinician that I can go and get this test in America and it takes a very long time to get, but this company are offering this sort of testing. I didn't realize that actually they're testing for a certain variant, which is actually in a population that I'm not a member of. So, people do need to be aware of the sort of test that these sort of companies provide.
I think they do need to be aware and read the small print, because the genes aren't everything, and the gene tests specifically, as Kevin mentioned, we have 3.3 billion letters of DNA, and double that up for mums and dads' contributions, so you go to 6 billion letters of DNA there, and we're testing for only 500,000 of them, and there's a brave bit in there that's not going to be covered by that. But interestingly, the BRCA1 and BRCA2 genes in the USA have been subject to patent until just the other day, with the company McCrea genetics, and that patent has been ruled unlawful by the Supreme Court of the US.
Yeah, we might talk about that in a bit.
Opened that up a bit.
The idea that the Supreme Court basically said you can't patent something that's naturally occurring. But let's move on, because we want to get to some questions.
So yes, the next one that we tested for then, well, this is just a little summary.
Yeah.
The next one was we wanted to look at your degree of risk for Parkinson's disease.
Okay.
Now, I think it's helpful in this context to sort of have an idea. I mean, what would you think? Don't look at the screen.
Okay.
What would you think the population risk of Parkinson's disease would be for a guy in your position, your age? You're perfectly fine, and the degree of risk that you would feel that you would have just without doing any gene test for Parkinson's. What would you normally put that at?
Very low. I don't know. I'd say.
Give me a percentage.
I have no idea. What the number of people who would have Parkinson's
Your
or might develop Parkinson's?
You're a numbers man, yeah.
I know 1 in 1,000.
Right, it's actually about 1.6%, so 16 people in 1,000 in the population. So it's a significant enough risk. Will develop Parkinson's disease if presumably they live long enough and everything else, other problems don't intervene before then. So 16 people per 1,000, 1.6%. Your own personal risk, and this is where the drum roll comes up, I am happy to say is actually a little bit lower than that. It is 1.4. So if there were 1,000 Jonathans, I'd only expect 14 of you guys to develop Parkinson's disease. So it's a little bit lower. But you can see the sort of the issue that we're dealing with here, that it's not a massive risk change. The risk started off relatively
so you could say,
small.
I mean, relative, it's quite small. Relative, it can be quite big sometimes. Sometimes you can see from these tests that, there's a 100% increase of your chance of getting Parkinson's, but that's still only a very small number of the population.
Yes, it's the difference between what we call absolute risk and relative risk. So relative risk is, if you see a lot of studies that are reported in the literature and particularly in the press, people say, your risk of hen's teeth disease is doubled if you drink 1 Heineken a day or something, and it maybe doubles from 0.001% to 0.002%. Is that really going to change very much in your life? It's not really. It might tell us something about the biology of the disease process, though. It might be useful for researchers looking at ways of treating the condition to know exactly how does this genetic factor affect the way the disease gets expressed. So that's useful information, but it's probably not useful for you as an individual, because if you've got the gene or you haven't got the gene, it doesn't make up a big lot of odds.
It's not going to be useful. Okay, so let's move on. What else?
So the next one then, the other real one that we wanted to really have a good look at was that you'd mentioned about your family history of the Alzheimer's disease, and obviously this is a family issue, and it's a concerning thing. And again, without looking at the screen, I mean, would you have a sort of ballpark notion in your head about how common this would be in the population?
I'd say it's quite, I would have thought quite high. I mean, I don't know. We hear about it all the time. It's one of the big diseases that we're worried about how we're going to deal with financially. I don't know. I really wouldn't. I'd say maybe, but I would say probably, more common than Parkinson's, right?
Yeah, I mean, it would be about 7.2% of the population will develop, if they live long enough, will develop Alzheimer's disease. So it's quite that's a pretty hefty number.
Yeah.
And obviously with the family history, that actually does increase your risk to a certain extent. But just looking at the genes and dealing with only the genes, we have been able to calculate that out. And next slide will indicate your own risk, hopefully, and it's gone down. So we're now down to 4.9%.
Wow.
But
so this is a specific gene that we know is a factor for Alzheimer's, and
this is based on the profiling of a number of different genes. There is one specific gene that we know is quite, or there are a number of genes, but there's one particular one that we see in the population that we know is quite strongly associated with Alzheimer's, and it's called the ApoE gene. And there's a particular variant of that gene that we know isn't present, but this profile is in your own DNA. But this profile is based on the information of a number of different interacting genetic areas, or loci as we call them, areas, locations within the genome. So your risk is down on purely genetic grounds on the DNA sample. The problem with that is that it's still there. It's not zero. It's still 4.9%. And the problem that we often run into is that people might be, for example, concerned about the mild or level of risk, say, in Parkinson's disease. We found that their risk of Parkinson's disease was 1.8 when the population is 1.7 or something.
Yeah.
But then they'd be reassured by an Alzheimer's disease risk of 4.9.
Like I am right now.
Yeah, like you are right now. But actually, the risk is still considerably higher than it was for the Parkinson's. But we know that to be just the way the statistics work, and also the way people react to the numbers that are given to them. So, I mean, how do you feel about that? What's your
well, I'm delighted. I mean, that's we've gone through all of them now. So,
so that's the ones that we were going to mention at this stage.
Okay. So, so in terms of bad news, it's all been relative, and with all the caveats involved, it's been good news. Well, that's good news, isn't it?
I think we're happy.
You haven't got like a surprise at the end, have you?
Well, no, really. I mean, we did look at other things as well, and I mean, well, yes, surprise. Well, one thing that I suppose would maybe be worth mentioning, because Kevin and I had a discussion about this earlier on with Tara and Lara. I hadn't really raised the rhyming consistency there, but the one area that the risk profile also looked at was the condition prostate cancer. Now we know prostate cancer is extremely common in the population, and in fact there's an argument that basically every male over the age of about 75 will have some prostate cancer cells in the prostate gland, and it's just the way it tends to work. And I hate to sort of be the bearer of bad news. It's not the bearer of bad news, actually. The background rate for that for prostate cancer is about 1 in 6 in the population. Your rate's up a little bit to 1 in 4. Still 75% chance that you'll never develop it, but it's one of those things that does sometimes increase to a certain extent. The question that becomes relevant, or the two questions that become relevant, one is that risk an accurate risk? And, I think both Kevin and I would be a little bit shaky on whether you can draw very many conclusions
because we're talking about a population level, and I'm one person, so whether or not that number is applicable to me is sort of iffy. Are you just trying to sugarcoat it, or is it actually what you feel?
No, I mean, this genuinely is the problem, and it's one of the issues I think that we would actually we would have about the whole thing of the whole health risk aspect, is that these numbers they do come with significant confidence intervals.
Yeah.
So they're giving you a sort of risk which is the best guess according to the data of various studies that have been done, and those studies themselves have got methodological problems sometimes, or they may be dealing with small sample sizes, and it's very difficult to get a clear result of those. So we're not going, I don't think, to be able to give you a clear risk, and certainly even with that level of risk change, you're not talking about something that will either, I think, give us evidence that we should say Jonathan should really be going and changing his behaviour in such and such a way, or should be receiving screening in such and such a time, or should do this or do that, or we should treat him this way or that way, or get him up on the operating table immediately and do things. There's nothing within any of those results. This is good news, by the way. This is the good news that we would regard as being clinically actionable. So I would be sending on your way. I'd say be happy and just eat. Well, I don't know.
Oh, wait, hang on a second. We'll come to a conclusion. So as my doctor, having looked at the results of this particular test, what do I do now?
What are you doing now?
What do I do now?
What are you doing now?
I'm eating
Keep doing
a bit of rubbish, exercising a bit, all the blah, blah, blah stuff that doctors tell you to do.
I mean, the bottom line
doing
is,
a bit of that.
you're not a smoker anymore, so we know that. We can take that off. You're not carrying too much weight, so we don't need to worry about that too much. You're probably getting plenty of exercise, I would like to think. And you're enjoying your life, and you've got a reasonably good diet. Why change that? This is the advice we would nearly give to everyone. So I think
as you were, says my doctor.
Pretty much.
All right.
At ease, Corporal.
Ladies and gentlemen, Shane McCrea. Well, if you're listening to this at home on the radio, this is the end of the program, but you can find out lots more about your likelihood of getting disease, these sort of genetic tests, and what it means to have this sort of information on our website. It's newstalk.ie/futureproof. We'll be back next Saturday with more. Thanks very much for listening. Okay, so I think we'll open up the floor for Q&A. So if you want to raise your hand, if anyone has a question, is the gentleman over here?
Ian, yeah, congratulations on your sort of sang-froid and getting that information cold. And as a result of it, I have a question for you. Given that what you had were a bunch of SNP tests, essentially, which are essentially clinically meaningless, would you now be willing to take the bigger step and have your genome sequenced?
I think I would. So SNPs are changes in those letters. So that's what we were talking about. So that's what this test looks at, the C to D or C to T or A to G. It's one of those single letters, and that's what this particular test of 23andMe looks at. I think I would. It's easy for me to say that because I don't have a history of Huntington's, thank God. I don't have a history of cystic fibrosis. There is one case of early onset breast cancer, but that's not something that is in my direct lineage. So yes, I would. If I did have that sort of history, I'm not so sure. But I suppose, just because I don't have those, I could find out something, a rare mutation that this sort of test doesn't look at. Isn't that right?
It's one of the issues with whole genome sequencing is you might go in for, say you do have a history of cystic fibrosis, and you would get that sequencing done. But if you sequence your whole genome, you can have what people call incidental findings. And then again, the genetic counselors have to be involved. You have to be informed to know, do you want to know those incidental findings? Do you have a right to know? Does the doctor have a right to tell you? And you could get a surprise that arises. It could even be a new mutation that's not even inherited that you didn't know about. And so there is potential there that you could get a surprise. But I think this is what's going to happen. The whole genome sequencing, the cost of that. In 2001, it cost $100 million to sequence a human genome, and it took years and years to do it. It can be done now in about 2 days for less than $1,000.
Yeah.
So, and that's going to keep on going down. So it's going to become a routine test. It's going to become a routine part of clinical genetic practice, and it may be the kind of thing that happens in this direct-to-consumer sort of way, almost with very little regulation in some places. Like the company in the States is not very highly regulated. Some other countries regulate this kind of stuff more.
Okay. I think one of the other issues with that, though, is that as we briefly mentioned there, the SNP tests that were done are presupposing that we have a lot of knowledge about those SNP tests and what they actually mean. And in fairness, a lot of those have been reasonably well validated to some extent, although the confidence intervals are wide.
And it's also difficult to get a scientist to ever say, that's exactly what happens.
Whole genome sequencing is going to pick up loads of things. For example, although you share a lot of your DNA with your mum and dad, there's going to be between about 50 and 100 mutations, new mutations within your DNA, new changes of those letter sequences that are not inherited, that happened in the egg cell or sperm cell that you were conceived from.
So I think that's an interesting kind of thing. So these mutations, a lot of the time, they're passed on from generation to generation. But then there are some changes which happen at the egg stage. It's a copying error, and they're brand new to me, and my parents never had
them.
That
happens every single generation. I mean, that's the basis actually for these SNP tests. The only reason these ones are ones that we test for is that they happened in some individual back thousands of years ago, usually, and they've got spread throughout the population. So there's a lot that we don't know about the effects that those are likely to have, and some of the studies that have been done so far on whole genome sequencing have picked up all sorts of mutations within genes that you would normally look at and say, oh my goodness, this person must be very, very unwell or have some major genetic syndrome, and actually they're walking around and they're perfectly fine. Yeah. And there's, you wouldn't pick them out, and everybody carries a certain number of these.
Yeah.
So, and we probably all carry quite a number of genes in the cystic fibrosis sense that we're carriers of that if we had them in a double dose would kill us stone dead. So. Yeah. We're okay. We have to tolerate a certain amount of genetic imperfection within our own genomes and also a little bit of genetic variability within the population.
Okay. Anybody else? Any other questions? This gentleman here in the shirt? Oh, this lady, this fellow here, this fellow there with the beard.
Considering the zeitgeist and the NSA thing that happened in the United States.
Yeah.
Is the biggest concern rather the fact that your DNA can be in some database in the US?
That's a good question, and I think it leads on to what are health insurance companies going to do with this? Because this is a procedure that right now, as Kevin says, costs $1,000 and it takes a while to do. In 5 to 10 years' time, it will probably be as regular as a checkup with a GP. And then there's a big question about the privacy. Well, you know, if you look like you might be likely to develop type 2 diabetes, well then, you know, are they going to bump up the prices? But more than that, like there's a real interesting thought experiment here. Say, for example, Obama runs for office again. I know he's on 2 terms, don't be pedantic. But if, say, he was to run again and he was up against someone like John McCain, for example, what if someone leaks his genome and says, look, you can see what a healthy person I am and that I'm fit to run this country, that I have genes associated with intelligence and so on and so forth. What about my opponent? You know, and you know, there's pressure then for you know, someone to reveal their genome information, because right now health information of a future president is very valid. You know, if someone's likely to have a heart attack, that, you know, the polls, people, you know, don't say it overtly, but, you know, secretly the polls, you know, plummet if someone has a heart attack in the middle of their primaries. You know.
Yeah, well, I mean, one of the things is that you know, already you know, insurance companies and so on have a lot of genetic information on you based on your medical family history. And in fact, a lot of this predictability from your actual genome sequence is in many cases not going to exceed that, or not going to exceed it very much. So many of these traits that we're talking about are partly genetic, but they're not completely genetic. So there will be some limits on that. Now, that said, the kind of probabilities that you're getting here are effectively actuarial. That's what insurance companies do. And actually, in Ireland and many other countries, there is a law that says insurance companies cannot use that genetic information to in any way discriminate against you or determine your premium that you will pay for health insurance. And I think it's true to say that they can't even use the information that you have had that test, or any test, to do that. So
a blow back to the HIV days, where you know, that was, if you had gone for HIV test, they would mark you down. Now they can't do that anymore.
That's right. Yeah. Yes, but it's, I mean, it's a big issue, the privacy thing, and who has a right to this information, and, you know, even technically, where is the information actually held, and can anyone hack into it and so on? Can we trust the government not to look at it if they're already looking at our emails and phone calls? So yeah, it's an interesting question.
Another question, the gentleman in the white shirt.
Sorry, I'm just trying to assess the beneficial information that could come from genetic testing in the following context. Okay, got a young child that's had a number of unexplained autoimmune diseases, one of them, the most obvious one, being diabetes, but another 3 or 4 organs that have been attacked by autoimmune reactions. I'm just wondering whether genetic testing could provide any information in terms of future risks or diagnostic information in terms of what's happened to date.
I mean, that would fall very much within the realms of genetic diagnostics, and would be the sort of scenario would be if a child has, say, an unexplained set of symptoms. Developmental delay would be a very common one, or congenital malformations, and perhaps we maybe don't know what the specific diagnosis is. You know, they'll come along to my clinic, and unfortunately this happens quite a lot. I can't immediately say this is such and such a condition. We can now carry out genetic testing, and until very recently, that has meant coming up with an idea: we should test for this condition, and then test that one condition, and then test another gene, then test another gene, and there's 23,000 genes. You could be a long time testing genes, waiting for each one to come back before you go on to another one. And we're now, just in the last couple of years, really getting into the stage where it is conceivable and possible to test all the genes in one big lump and find what comes out of that. That's called exome sequencing, which is just looking at the protein coding genes, or genome sequencing, which is all the genes. And that's already being brought into clinical practice largely in a research setting in the UK and Ireland, but also being rapidly moved into diagnostic settings. So
if someone has a son or daughter that is showing some sort of autoimmune response that isn't normal, or the normal tests don't come through, do people get referred to you then to see if there's a genetic basis to it?
Potentially. There's a research study at the moment going on. It's based in Cambridge, and both Dublin and Belfast are currently part of that research study. It's a study called the DDD study, and it's using the latest genetic technology in exome sequencing and in analysing the data that comes back out of that to try and find the causes of disabilities, birth defects, and various other problems, particularly in children. And that's a very exciting study. Cambridge was one of the main centres that sequenced the human genome back when that was released, and they've got a very well-appointed genome centre there. So this is a research study that's trying to pull that together and trying to put all this information into one place. We're going in pretty much blind because a lot of the genes for these conditions aren't known yet. So if you find a change in a particular gene, you don't necessarily know that is causing the problem in that child. There's 23,000 genes. We maybe know what 5,000 or so of them do.
But this is a good justification for getting more and more of the population to get their genome, because the more people that we have who get their genome sequenced, then the more we can find these sort of links.
Well, if the data is available to the researchers, and at the moment it seems to be that the best way of doing this is within a research setting where there's a very tightly governed and looked at research hypothesis that's being investigated. So we're asking a specific question, and going to try and find the answer. But there's no doubt that genome sequencing is revolutionising molecular diagnostics at the minute and will hold out a lot of hope for children with these problems and speed up diagnosis.
Kevin?
Yeah, I think one of the other aspects of that is if you do get a genetic diagnosis, then you can ask, was this a new mutation that arose, or was it inherited from mom or dad? And if it's inherited from mom or dad, then you can say there's a certain risk to the offspring to additional offspring of getting that. And if they get it, then if in the population of people who carry that mutation, you could say 80% of them develop this condition, or 60% or 100%. So that is useful information in terms of predictions and the possibility of making reproductive choices for people as well. So it goes beyond the initial diagnosis in the individual.
And even having a name for a genetic condition is often a very powerful part of not exactly the treatment but the management of that child. Yeah. So a lot of parents would have a child with a severe problem, and they've maybe struggled on for years back and forward with various clinics, and everybody knows what the problem is with the child, or rather what the medical problems are, but they just can't pin it down to what the actual cause is and what the name is. And sometimes if you can come along and say this is Coffin-Siris syndrome, or this is such and such a syndrome or a condition, then that can be a huge weight off the mind of the parents, and also allow them then to maybe contact other families who are in exactly the same situation, share information, and even then feed back into the whole research effort in managing those children. And there are quite a number of examples where the parents have driven the management forward and have found out things. This works with my kid with this condition, and that's been very, very helpful.
This gentleman here in the beard who bought me a caramel square, we'll get to him in a sec. Trina.
I was just wondering, as you share so much of your genetic information with your family, how much input they have on your choice to go public with your genetic information. And if you were to do a whole genome sequence, you would be sharing a lot of their information. So what do they have to say?
Dad, do you want to answer that? Because I said, my dad and my mum, I love them very much. They're very doting. Having said that, I kind of tell them I'm doing something, and I go, oh yeah, okay. Did you fully grasp what was going on, or did you kind of go, oh, that sounds nice, son?
Well, I didn't know that you were going to do this.
I told you I was going to do this.
I know.
I sat you down, I said, it's really important you understand I'm doing this.
You did not.
That's the Alzheimer's.
No, I didn't know you were going to do it until just recently. So I have no objection to what you would find out. It's interesting, very interesting to find out the various things that are there. But I have no objection to doing the whole lot.
Yeah. Would you not? You wouldn't mind if I looked at the entire genome?
No. Well, I feel I'm kind of set now in my whatever I have, and
old people think that way.
I'll find out if there's anything else coming. I'll find out in due course. I don't really mind either way. If it's coming along, I'll.
You've no interest in finding out, there are some genes that are associated with Alzheimer's, but with Granny, your mum,
well, I
you wouldn't like to know...
I think I probably have a slightly higher risk of Alzheimer's, and I do occasionally sort of try and remember the names of capitals of American states and things, just to make sure it's, just to check whether it's coming on or not. But so far, so good.
Shut up. He actually does know all the names of the capitals of the
Wisconsin.
Mom?
Madison.
Mom.
Whoa.
Show off. This is my show. Mum.
I think it's very,
Use the microphone, Mum.
sorry. I think it's very important to think about, if you get a certain amount of information about some disease that you may or may not, may possibly get, that you have a very high risk of, I think it depends on the sort of person you are. If, for instance, you're prone to depression or negativity or have had any kind of depression in your life, I think it could be a very important factor in it.
Yeah.
Because, for instance, if you were told today that you had a very high risk of getting Alzheimer's, if you were a depressive sort of person or a negative person in any way, how would that affect you? And would it change your life forever?
But, I mean,
getting this, a huge piece of information about you, would you begin then to think about it a lot? And would it take you over in the end? Could it possibly take you over?
That's a good question. I think, Shane, when it comes to delivering the sort of information, does the type of personality you have affect how you react to it?
There has been a lot of discussion about how results and things should be given, and there's a general feeling that people have the right to know their information, and sometimes there's a perception that we won't tell them certain things, or we ask them, particularly in the Huntington's disease testing, because it's such a devastating diagnosis potentially, that there's sometimes a perception among some people we're putting barriers in the way of them accessing that information, and we try to do it the other way around, that the course of the genetic counselling process is to try and give the person the skills, if you like, to handle the information that's coming on. But that's absolutely correct. This can be a very big blow to people, and what we need people to do is to think very, very deeply about whether this is information that they feel that they can handle, and whether it is something that's going to take over their life, because it really shouldn't take over their life, because the sort of information we're getting from these tests isn't something that really puts the rate up.
No, but I suppose for other tests it might do, but
the other things might.
The irony, I suppose, is that there are also genes for depression. So that you might be genetically predisposed. Thank you. Predisposed to being depressed, and then get this result about your genes, and then get depressed about that. Any other questions? Okay, this gentleman with the caramel square.
I hope you like the square.
It's delicious.
So my question is about the future of this technology. I'm sure a lot of people here have seen the film Gattaca and the kind of testing that's done. And in one scene, somebody takes a potential mate's hair and gets it tested.
Yeah.
And we see a lot of genetic sculpting. So just like you guys to discuss the ethics and the potential future. Are we going to see this kind of sculpted society where everything is, there's no flaws in anybody left?
I was talking about this with Kevin, and this is a really interesting area, because right now we can edit one of those letters, 6 billion letters, so we can edit and fix that letter in an embryo. And so there's an ethics question as to do we do that? In a mouse embryo, we could do that. Yeah, in a mouse embryo. No, but we technically could. We technically have the possibility of doing that. And the question is, what conditions do we fix in an embryo, and where do we stop?
Yeah, and I think, actually, even before that, the Gattaca scenario is one where they're not tinkering with the genome. All they're doing is selecting things. And the way that they do that is with basically what's now called pre-implantation screening or diagnosis. So they in vitro fertilize a bunch of embryos, look at their genetic profiles, see which ones are high risk for various things, and then implant the ones that are low risk or that have more desirable predicted traits, whatever they are. And in fact, that happens now already. So people do pre-implantation diagnosis for disease screening if they know that they carry disease alleles like the Huntington's, like cystic fibrosis and so on. And that goes on. It also, interestingly, in some places happens for other things. So in the States, for example, where it's not as tightly regulated, you can pre-screen for sex of an embryo and make a decision on that basis. And you can screen for a few other things. In fact, there are even cases of people screening and selecting for the presence of a genetic variant that will cause what society calls a disorder, but what they don't see as a disorder. And that would be cases of deaf parents, in some cases selecting embryos that will be deaf, or parents with dwarfism selecting embryos that will also have dwarfism. So,
wow.
So there's a whole range of possibilities there. And I guess the Gattaca scenario is that we will be able to predict all kinds of things, and people will be able to select for all kinds of things, like IQ, for example.
Hair curl, red hair,
curl,
red, joking aside.
Sure, left-handedness, you can think of all kinds of things. And so it's an interesting scenario. I think the good thing is that no matter how good the genetic prediction gets, for most things there's still going to be some variability left over, because identical twins are not going to be completely identical for lots of traits. But the other thing is that there's our knowledge base at this moment in terms of which genetic variants predict which traits is really pretty rudimentary. Okay. And I don't know if it came through from the profile that you got, that the match and the differences in health risks and so on weren't that great for most of these conditions. And I think that's likely to continue to be the case for some period into the future.
Yeah.
And there's the interaction between the different genes. They all interact. So you won't even be able to look at one single nucleotide polymorphism, one of these SNPs, these variants, and be able to say that this will definitely cause a certain thing, because it may be influenced by another one that's somewhere else. So if you've got, even among the 500,000 that we have, the interactions between those are so potentially vast that the number is just way off the scale, and there aren't going to be enough people born in the world to look at every single possible interaction.
Not yet.
Of those. Well,
not ever.
we may need to move off world, I think, if that's going to be the case. Yeah. But we're limited by our knowledge.
Yeah, you can think about it like getting. We were talking earlier in the casino that you can get a look at the hand that
at
you're
11
dealt.
o'clock in the morning.
Right. So basically, these tests give you a look at the hand that you're dealt. This test gives you a look at maybe one card. Okay, the whole genome sequencing shows you your whole hand, but really we don't understand the rules of the game that we're playing. So even if you see the whole hand, if you get all that information, you still don't really know what the outcome is. So we're still learning the rules of the game, scientifically speaking, in terms of how the genotype influences the phenotype.
Okay, I think we'll end with a final question, and that is, knowing what you know after hearing this evening, if you had the chance of finding out that you were likely to get a fatal disease, would you want to know? Raise your hands. I think it's about the same. Anybody change their mind? 1, 2 people. Okay. Job done. Ladies and gentlemen, thank you very much. I'd like to say a huge thank you to Shane McKee, Kevin Mitchell, and Tara O'Neill for taking part in our panel. Shimmer Biosensors, who were providing the sensor there. You could see how freaked out I was. Massive thanks to our composer, Mark Hennessy. He also composed some other music for us. He's absolutely fantastic. And check it out, facebook.com/Mark Hennessy Music. Thanks to the Science Gallery, who were brilliant putting this together. Thank you and good evening. See you later. Bye-bye.